Welcome to Amino Lab channel. My name is Fidan and we're starting a new series called Zero to Genetic Engineering Hero, where we're going to explore the book Zero to Genetic Engineering Hero and we're going to do the corresponding kits. For the kits and the book, please check out the description box below. Today is our first episode, the first step, the first chapter, and we're going to learn about the basics of genetics. Then without further ado, let's get started. We're familiar with computer. It has many parts: keyboard, memory, screen, hard drive, and many other. As you may have noticed, they can be made of plastic, glass, silicon, steel, or iron. Just like that, human bodies are made of cells that are a bit more complex. Cells are fundamental building units of living organisms, and they are made of proteins, carbohydrates, lipids, and nucleic acids, which are the focus of today's video. One of the crucial nucleic acids in our cells is DNA, DEOXYRIBO acid, which contains all the necessary information to build a specific organism. DNA is a microscopic string of tiny molecules called nucleotides, and their order makes a language that the cell knows how to read. Imagine DNA as a hard drive of the computer. It holds the information and helps the whole computer to function. By changing the dots and dash information on the hard disk, the computer behaves differently, which is the same in DNA. Any change in the order of nucleotides will result in a change in execution of the DNA. There are a variety of factors causing these changes, like ultraviolet light, radiation, chemicals, viruses, and etc. And exactly DNA's ability to change has become the basis of evolution. Evolution is a change in the characteristic of organisms over time, which was closely examined and discovered by Charles Darwin. It made every organism unique and created an amazing variety in fauna and flora. Did you know that 5 to 8% of human DNA is made of viruses that once infected our ancestors? One of them is called HIV, human immunodeficiency virus. And it shows that human DNA hasn't been human solely from the beginning, but rather an accumulation of different organisms. If you like this fact, don't forget to give it a thumbs up. The founder of genetics and biotechnology is known as Gregor Mendel, who determined how traits are passed from one generation to the next without even knowing about DNA itself. His famous pea plants experiment brought light to the inheritance of specific traits over one generation to the next and re-emergence of other traits for other generations. When he crossed the yellow and green peas, the third generation of offspring were all yellow. But when he crossed those with each other, he found 1/4 of the peas were green. He started determining some genetic rules and coined terms of recessive and dominant genes. Yellow that emerged at the beginning were dominant, and the green that re-emerged later was recessive. And the combination of Darwinian evolution and Mendelian genetics created today's unified framework to understand biology. If you think about it, different colors of peas are also due to changes that happen to the peas. And you might ask, was that for the best? Are changes good for organisms? Changes in DNA are called mutations, and they can be good, bad, or neither, but it's not that easy to say right away which one it is. Sometimes they cause diseases and they're obviously bad, but sometimes they're good during one time, bad during a different, or neither on a different period, depending on the environment. If an organism has a beneficial mutation, it survives more those who didn't, and they reproduce and pass their beneficial mutation to their offspring and so on. That's called natural selection. Imagine a group of mice living on a dark rock. Some mice due to mutation have white fur and the rest has black. As the black fur camouflages the mice, the birds can't see the black ones and instead they eat the white mice. Over time, only black mice survive, reproduce, and become the majority living on that rock. That mutation due to environment wasn't beneficial. However, during winter times, the opposite happens. The black mice get cold and the white ones survive. In that case, the mutation was beneficial. As you can see, like many other things in the universe, the impact of the mutation is also relative and depends on the environment. Now you might ask, what is DNA made of and how those changes occur? DNA is made of 4 chemical building blocks called nucleotides, attached in various orders forming the DNA string. Each nucleotide is made of a molecule called phosphate, a nitrogenous base, and deoxyribose sugar. Phosphate is a molecule attached to 4 oxygen atoms. There are 4 nitrogenous bases in DNA: guanine, thymine, adenine, and cytosine. Erwin Chaga found that the rule guanine is always with cytosine and thymine is always with adenine. Gene and AT are also so-called Watson-Crick pairs. Deoxyribose is a sugar ring that is the D in DNA, and together with a phosphate, they form the sugar-phosphate backbone. When 2 strands of DNA come together, they form the structure called double helix, which looks like a spiral staircase. Or imagine a ladder. The sugar phosphate is the outside of the ladder, and the bases are the ladder rungs. The outside of the ladder is to maintain the structure so that the special machines called enzymes can read the bases easily. In addition, to help the cell to read the material, the bases are connected with so-called hydrogen bonds, and they're easy to unzip like the zipper on a jacket to read and then to zip back to protect for the future. Now let's go over things we learned today. Cells are fundamental building units of living organisms. DNA is deoxyribonucleic acid and contains the necessary information to build those organisms. Gregor Mendel is the founding father of genetics, and together with Darwin, they established today's framework to understand biology, what mutations are and how they can be good, bad, or neither depending on the environment. And we learned about structure of DNA being double helix and about the Watson-Crick pairs. Thank you so much for watching, and please like, comment, and subscribe if you found this video helpful, and don't forget to press the bell button so you're always up to date with science. You can check out the description box for more resources. And young scientists, until the next video, let us know in the comments what about DNA has fascinated, wondered, or confused you the most. See you in the next video where we're going to explore application of genetic engineering in real life, and in the next video we're going to do the DNA extraction.